Hello everyone, welcome back to this channel. In this video, we'll be completing the biological molecules part of the A-level revision AQA, International AQA syllabus. Also, I believe this video must be useful for Igcse and OCR. Okay, so the structure of the carbohydrates, lipids, and proteins were done in the first video. In this second video, we'll be doing the test of the proteins, the enzymes, the structure of the DNA and the RNA, the DNA replication, the ATP, the water, and the inorganic ions. So starting with the inorganic ions occur in solution in the cytoplasm and body fluid of organisms, some in high concentrations and others in very low concentrations. Some of the essential ions include the hydrogen ions, the iron ions, the sodium ions, the phosphate ions. The hydrogen ions, which determine the pH of substances such as blood. The higher the concentration of hydrogen ions, the lower the pH. Iron ions are a component of hemoglobin, which is an oxygen-carrying molecule in red blood cells. Sodium ions are involved in co-transport of glucose and amino acids. Phosphate ions are a component of DNA and ATP. What is ATP? ATP is adenosine triphosphate, and it's a nucleotide derivative and consists of ribose, adenine, and 3 phosphate groups, as you see in the diagram below. Sorry, to the right. Basically, energy is released. Energy is released when ATP is hydrolyzed to form ADP and the phosphate molecule. This process is catalyzed by ATP hydrolase. Again, energy is released when ATP is hydrolyzed to form ADP and the phosphate molecule. This process is catalyzed by ATP hydrolase, which is the enzyme. The energy comes from the bonds between the phosphate molecules, and these bonds are very unstable and thus have a low activation energy. The breaking of these is quick and releases a considerable amount of energy. The inorganic phosphate can be used to phosphorylate other compounds, as a result, making them more reactive. Condensation of ADP and inorganic phosphate catalyzed by ATP synthase produces ATP during the photosynthesis and respiration. We have some properties of ATP. ATP is an immediate source of energy. ATP isn't stored. ATP is used in variety of different ways, which include the metabolic processes, movement, active transport, secretion, and activation of molecules. We also said that ATP is an immediate source of energy and is more desirable to use than glucose, as ATP can be broken down in a single step to release a manageable quantity of energy. Lastly, ATP isn't stored in large quantities, as it can easily be reformed from ADP in seconds. Then we're going to go to the water. What do you know about water? We know that water is a very important molecule, which is a major component of cells. For instance, we need to know that water is a polar molecule. Water is a polar molecule due to the uneven distribution of charge within the molecule. The oxygen atom attracts electrons a bit more strongly than the hydrogen atoms. So the unequal sharing of electrons gives the water molecule a slightly negative charge near its oxygen atom and slightly positive charge near its hydrogen atom. Also, water is a metabolite in metabolic reactions such as condensation and hydrolysis, which are used in forming and breaking of chemical bonds. If you do not know about them, please go check the other video. Where condensation is for joining, hydrolysis is for breaking. That's a quick introduction about them. Well, that's all you need to know, yani. It's a solvent. Water is a solvent, allowing gases to readily diffuse, as well as enzymes and waste products, for example, ammonia and urea. Also, water has a high heat specific capacity. This is because water molecules stick together with hydrogen bonds, meaning that a lot of energy is required to break these bonds. This helps to minimize temperature fluctuations in living things before it acts as a buffer. Also, hydrogen bonding means that it requires a lot of energy to evaporate 1 gram of water. Therefore, water has a relatively large latitude of vaporization, meaning evaporation of water provides a cooling effect with little water loss, for example, sweating. The strong cohesion between molecules enables effective transport of water in tube-like xylem, and the strong cohesion supports column of water. As a result of strong cohesion, the surface tension at the water-air boundary is high. That's completely great. Now we're done with carbohydrates, lipids, proteins, and water. But we haven't done enzymes. Do you know what enzymes is? Basically, enzymes are biological catalysts, which means that enzymes increase the rate of reaction by lowering the activation energy of the reaction they catalyze without being changed. They are 3D tertiary structure globular proteins whose shape is determined by the primary sequence of amino acids. This indicates the enzymes are actually globular 3D tertiary proteins. Now, here in the enzyme, there is the active site. The active site is an area of the enzyme that is made up of only a few amino acids and forms a small depression in the overall enzyme. The molecule that the enzyme acts upon is called the substrate. Enzymes are specific to substrates and they bind to, meaning that only one type of substrate fits into the active site. Fits to the active site of the enzyme. When the enzyme and substrate bind, they form an enzyme-substrate complex, and the structure of the enzyme is altered so that the active site of the enzyme fits around the substrate, which is called the induced fit model. There are some factors that affect the rate of enzyme-controlled reactions, from temperature, pH, enzyme concentration, substrate concentration, concentration of competitive reversible inhibitors, and concentration of non-competitive reversible inhibitors. I know you've known about temperature, but let's revise. Temperature, basically, the rate of reaction increases up to the optimum temperature as the kinetic energy of the enzyme increase. Above the optimum temperature, rate of reaction decrease beyond the optimum temperature as the enzyme becomes denatured. For the pH of a solution is the measure of the hydrogen ion concentration, and the pH of a solution can be calculated using the following formula. pH is equal to negative log 10 H plus and hydrogen ion concentration of 1 times 10 to the power of negative 9, therefore has a pH of 9. pH affects the enzyme's shape as it can disrupt the bonds in the tertiary structure of the enzyme. Like temperature, all enzymes work at a different optimum pHs. For example, pepsin in the stomach works in very acidic conditions. Enzyme concentration. The rate of reaction increases as enzyme concentration increases and as there are more active sites for substrate to bind to. However, increasing the enzyme concentration beyond a certain point has no effect on the rate of reaction as there are more active sites than substrates, so substrate concentration becomes the limiting factor. Going to the part after, which is basically the substrate concentration. As concentration of substrate increases, the rate of reaction increases as more enzyme substrate complexes are formed. However, beyond a certain point, the rate of reaction no longer increases as enzyme concentration becomes the limiting factor. Now we go after the concentration of competitive reversible inhibitors. As concentration of competitive reversible inhibitor increase, rate of reaction decrease as the active sites are temporarily blocked by inhibitors so substrate cannot bind to them. The non-competitive, basically they non-compete. So as the concentration on non-competitive reversible inhibitor increase, rate of reaction decrease as the shape of the enzyme, not the active site, is altered by the inhibitors. They compete on the enzyme shape. So let's just finish the enzyme part, which basically do the test of the protein. Finish the protein part, which is the test for proteins. The Bayer test. The Bayer test can be used to test for the presence of peptide bonds in a protein and therefore to detect proteins in food. The test is carried out as follows. First of all, place the sample to be tested in a test tube and add an equal volume of sodium hydroxide at room temperature. Then add few drops of very dilute 0.05% copper to sulfate solution and mix gently. A purple coloration indicates the presence of a peptide bond and hence a protein. A negative result would mean the solution remains blue. This is the test of the protein and it's called a Bayer test. Now we will be doing the structure of the DNA and RNA and the DNA replication to be finished with the first part of the unit 1. Not first part of unit 1. Biological molecules. Second part of the biological molecules. This is the last part, guys. Structure of DNA and RNA and how the DNA is replicated. Oopsie, let's go back. So, starting with the DNA and the RNA and these kind of stuff. What do you know about them and what are the similarities? First similarity is that both DNA and RNA carry information. For instance, the DNA holds the genetic information, whereas the RNA transfers this genetic information from the DNA to the ribosomes for protein synthesis. Here, the ribosomes are formed of the ribosomal RNA and proteins. Both deoxyribonucleic acid, which is basically the DNA, and the ribonucleic acid, which is the RNA, are polymers of the nucleotides. So the nucleotides is the monomer for them, or what we say by the So the nucleotides consist of pentose, which is a 5-carbon sugar, and a nitrogen-containing organic base, as well as a phosphate group, as you see in the diagram as the right. Pentose sugar with a base and a phosphate. The components of the DNA nucleotide are deoxyribose sugar, phosphate group, and one of the nitrogen-containing organic bases, adenine, cytosine, and guanine or thymine. For the RNA, basically they have ribose sugar. First one has deoxyribose from its name, deoxyribonucleic. Second one, RNA, has ribose from its name, RNA. Yeah, ribosomal and ribonucleic. Okay, and a phosphate group, and one of the nitrogen containing organic bases, adenine, cytosine, guanine, or uracil. Okay, as well. So the nucleotides join together by phosphodiester bonds formed in condensation reaction. So it formed in condensation reaction by a bond called forms phosphodiester bond. The result is a dinucleotide which join to form polynucleotide. The bond forms between the deoxyribose sugar of one nucleotide and the phosphate group of another. A DNA molecule is a double helix composed of two polynucleotides joined together by hydrogen bonds between complementary bases, whereas RNA is a relatively short polynucleotide chain. Base pairing nitrogen-containing organic bases occurs as follows. The adenine always pairs with thymine, and the guanine always pair with cytosine. You can say at garage. A, T, adenine, and thymine. Guanine, garage looks like each other. The C looks like the G, so they bind together. Bind together. Bind was like a formation between bind and join, right? So the DNA is a stable molecule for the following reasons. Two reasons. First of all, the phosphodiester backbone protects the more chemically reactive nitrogen-containing organic bases inside the double helix, and that the hydrogen bonds form bridges between the phosphodiester uprights, as there are 3 hydrogen bonds between guanine and cytosine, rather than 2 for adenine and thymine. A higher proportion of CG pairing makes DNA more stable. Again, so the hydrogen bonds form between bridges between the 5 phosphodiester uprights, as there are 3 hydrogen bonds between guanine and cytosine, rather than 2 between adenine and thymine. But the more the CG, the more the DNA is stable. Then we have the DNA replication. We're going to be totally done with whatever is up, totally done with whatever is down. Only DNA replication is left. So I want you to look at it. Tell me, what do you see? What do you know, and what are you looking forward to know, and what did you understand after reading it? Okay, welcome back. So, the DNA replication. The semi-conservative replication of the DNA ensures genetic continuity between generations of cells, meaning that genetic information is passed on from one generation to the next. From one to the next, and from next to the one, right? The steps of semi-conservative replication of the DNA are as following. First of all, an enzyme called the DNA helicase causes the 2 strands of the DNA to separate, breaking the hydrogen bonds between the complementary bases. One of the strands is used as the template, and complementary base pairing occurs between the template and the free nucleotides. So once activated, nucleotides are bound, and the enzyme DNA polymerase joins them together by forming the phosphodiester bond. This result is that the 2 identical strands of DNA are formed. This is the end of this video. We're currently done with revising all of the biological molecules using PMT notes. I want you to know that this is not the end of the DNA. We still have a completely separate chapter for the genetic information, variation, and relationship between organisms, as well as that we have another chapter of genetics, as well as the population, evolution, etc., right? So do not worry, we still have lots about DNA. That's a quick introduction of what we need to know for this part. Have a nice day. Thank you very much for watching, and I wish this video was useful for you. Do not forget to watch the other vision videos, which we will be using other notes to be make sure that everything is 100% sure and 100% stable in your mind. Have a nice day.